Hi, this is the showcase presentation for the plant phenotyping team. The overview for this showcase presentation, we will cover a few different things. First, we will talk about the company, and we will introduce our project at a high level, then the stakeholder and the team member, the problem that we need to solve, and the opportunities our project provides to solve the problem, the solution that we built over the last 9 months, and then the results and highlight of our projects, and some lessons that we learned during the whole process. For the company, plant phenotyping at the University of Nebraska is accelerated by the use of an automated greenhouse, which is located at the Nebraska Innovation Campus. This system includes greenhouse base and an automated conveyor belt with 672 plants capacity. The system is fully automated and environmentally controlled, with automated watering and weighing stations, and 4 chambers equipped with cameras capable of collecting images in the visible, infrared, fluorescent, and hyperspectral range of each plant. And it's able to grow and photograph hundreds of plants simultaneously over the course of several months. And the problem is that processing the photographs to extract plant traces is computationally intensive if it's done manually, because hundreds of plants can be photographed every day over the course of an experiment. Therefore, an automated workflow is needed, and our team was required to create the automated workflow to reduce the running time and to improve the processing efficiency. For the stakeholder, our stakeholders include members in HCC, the Holland Computing Center, and the Schnabel Lab, which emphasizes on plant genomics and phenomics. Here are our 4 stakeholders. Dr. Derek Weitzel, who is a software engineer at HCC and also a research assistant professor at the CSE department. Dr. Hongfen Yu, who is an interim director at HCC and associate professor at the CSD department. Chen Yong Miao, who is a sponsor assistant and also a PhD student at the Schnabel Lab. And at last, Dr. James Schnabel, who is a supervisor of Schnabel Lab and associate professor at the Department of Agronomy and Horticulture. Then here's our team, our squad lead and product manager Ying Chao He, a senior in computer science, and myself, Zheng Hui Su, the development manager, and also a senior in computer science. And here's our 3 developers, Colin Corman, a senior in computer science, Philip Yuan, a junior in software engineering, and Evan Yu, and also a senior in computer science. Then here's our tribe lead, Dr. Bourne, and our project manager, Manny, and at last our coach, Lamin.
The problem initially faced by the Holland Computing Center is their need to manage and interpret terabytes of photographs of corn and sorghum. They wish to find a way to better manipulate that data to allow them to extract the traits of corn and sorghum for the series of images that showcase their growth. To do this, we use the Pegasus Workflow Manager to integrate the plant phenotyping modules into a seamless workflow. The workflow is then executed on an HPC cluster managed by Pegasus to improve the phenotyping model processing speed. Further, we developed a data compression library to reduce the number of files in the dataset to decrease the storage requirements for those terabytes of data. This allowed us to access different aspects of the corn and sorghum, such as their height and width of their inflorescence, the height of their stem, and the height of the plant as a whole. To give an overview of the product we have created for the Holland Computing Center and the Schnabel Labs, we created the above graphic. This shows the 5 main phases of the workflow that we integrated over the span of the last year. Initially, we accessed our compressed zip files of the hyperspectral images, then we select the desired images to be unzipped and interpreted. Then in phase 2, we unzip those selected images and convert them to a single NumPy array using machine learning to isolate their organs. Next, in phase 3, we convert the NumPy array to a 3D array, which colors the defined organ groups by assigning them RGB values. This then returns a completed predicted image to phase 4, where we use image processing for this prediction to get the desired dimensions of the organs. Once we have these measurements, we pass them to phase 5, where we add them to their respective CSV file, creating a record of their growth of the organs over time. Here we can see an example of the image processing that is used to isolate the organs for trait extraction. The starting step is to select a region of interest to establish an area where our data is going to be found. Next, we erode the noise from the image, removing unnecessary areas of selection which were improperly colored in the prediction process. Then, we dilate these more concentrated areas to allow for reselection of the erroneously filtered areas. We then refill the initial color for that area so that we can perform the edge detection on the selection. Edge detection allows us to get solely the perimeter of the isolated organ. Since our goal is to attain just the width and height of the organ, the north, east, south, and westmost positions are only needed to calculate those traits. Next, we clean the selection once more using dilation and erosion to ensure the region of selection has the highest accuracy to reality. Finally, we fill in the color once more, and we complete the trait extraction by drawing a box around the image and using a pixel to centimeter ratio computation to collect their dimensions in reality. Data for the trait extraction of each organ for a single plant's growth. The top shows growth of the inflorescence width, the next the inflorescence height, then the stem height, and finally the plant's total height over the span of time. These graphs provide a great visual for understanding the value of the workflow system. They allow the user to track the growth trends of these organs during the plant's growth, allowing them to make inferences about the growth conditions' effect on the plant's development, which they can then use to find areas to look at more closely and conduct further research on. The workflow was created using Pegasus, a workflow management system that represents each of these individual jobs as nodes on a directed acyclic graph. Pegasus converts several small detailed tasks into a singular, simple, and less computationally expensive task. Then, we implemented Pegasus clustering, which groups together independent jobs into one large job. This helps to improve the runtimes and wait queues on the HCC servers, allowing for other users to have greater access for job submission. In addition, a new compression workflow was then created separately from the prediction workflow, allowing for the initial image sets to be compressed to zip files before storage. This only moderately reduced the size of the Schnabel data, as the initial images for each plant were already held as PNG files, which are already compressed. However, with datasets as large as the ones used in this project, that small percentage compression can be a significant amount given the terabytes of data. We also captured and measured several traits for corn and sorghum, allowing that data to be displayed and used for scientific research. A few highlights we had were in our ability to plan and ensure we always had spare time for the evolving needs of the HCC and Schnabel lab. We created a strong testing strategy before we began development, so we were able to create a product that fulfilled our sponsors' needs. Communication with the sponsors was key for these successes, as constant contact allowed for continued specification of requirements and goals. Finally, evenly distributing tasks among team members was quintessential to discuss successes, as tasks were then able to be completed before their due dates. The Plant Phenotyping Senior Design Group would like to extend our appreciation and thanks to our sponsors at the Holland Computing Center. Their expertise and advice was incredibly helpful during our development process. We hope that everybody stays safe and stays healthy.